Imagine, your mom has baked a delicious cake, and you and your sibling are eagerly waiting to dig in. But wait, your mom wants to make sure both of you get the same amount, so she takes a knife and divides the cake into two absolutely identical halves. Each half has the same frosting, the same number of sprinkles. It's as if she's performing a cake mitosis, ensuring that each of you gets an equal share of deliciousness. This is what exactly happens at the end of the cell cycle. The cell that you refer to as a parent cell goes through a series of steps to pair itself for the time where it has to be divided into two identical halves. We call these the daughter cells. The process of mitosis can be broken down into four subphases: prophase, metaphase, anaphase, and telophase. Then after mitosis has just ended, the cell splits into two. Now let's delve into how each of these subphases contribute to the process of mitosis. Prophase. The chromosomes that are loosely hanging around inside the cell's DNA, they become more condensed. This happens because later they have to split apart. So to ensure that each daughter cell receives an accurate and complete copy of the genetic material, the chromosomes have to be in a condensed and organized manner. The second thing that happens is that the nuclear envelope, aka the nuclear membrane, breaks down, releasing the chromosome as a result. This is necessary because it allows the chromosomes to split up easily in the further steps without any obstacle coming in between. Last but not the least, the two centrosomes migrate to the opposite ends of the cell. Don't worry, we're going to discuss what happens just in a moment. Metaphase. Now the centrosomes, they are the master of organization. They send out these long ropes to adjust the chromosomes in the center. These structures are called spindle fibers, and their function is to line up these chromosomes in the middle at the metaphase plate. Anaphase. In this step, I like to refer to it as a tug of war between the chromosomes and the spindle fibers. The spindle fibers that grabbed hold of the chromosomes pull them apart with all their might towards the opposite ends of the cell. Each is now its own chromosome. Now, just to clarify, I know it's a bit confusing that a duplicated version is a chromosome and a single version is also referred to as a chromosome, but what we actually do is count the number of centromeres. That's the centromere over here. One centromere corresponds to a single chromosome. So when they're duplicated, they have one centromere, meaning that one chromosome, and when they're pulled apart, they again have one centromere each. So this is also called a chromosome, and so does that one. Back to our anaphase, so yeah, that's just a tug of war. Moving on to the last phase of mitosis, we have telophase. Now the chromosomes get back to that loose spaghetti-like structure, that is, they begin to decondense. The nuclear membrane starts to form around each of the new sets of chromosomes. We're done with preparing all the essentials that the new daughter cells will require for their own survival, growth, and development. And now for the grand finale of the mitosis show. The cell membrane starts pinching in and the cell divides into two separate parts. We now have two identical daughter cells, ready to take on the world. It's a magical moment of cell replication, bringing us more of those amazing building blocks that keep our bodies running smoothly.